Welcome to this live session. This is the first live session for our course on dynamical models in biology. So I hope the learners will come and will have some interactions. So if any of the attendees has any question, you can post the question. We will start the session. If any, it will be nice if some of you can post your name and the background that you have. So what subject you are studying and those things, if you can post, it will be helpful to start the discussion. Okay, if there is no question here right now, let us start with some basic discussion, right? So this course is a course on dynamical systems, right? And building dynamical models for biological systems. Particularly, what we are doing, we are focusing on models which are related to cell cycle, cell signaling, transcription, translation, and all those things. And if you have looked into the curriculum of this course, you must have noticed that although it is a mathematical biology course, still what we are trying to do is that we are trying to make it more graphical so that it is accessible to most of the people, right? Most of the people who are coming from mostly from biology background. Obviously, you need some amount of knowledge of calculus to handle this course. Otherwise, most of the things that we will study in this course, we really do not need to go in details of math, right? Mostly we try to capture the essence, the ideas which you can use. This is an elementary course, so you can, if you learn this concept, then you can dig into more or advanced courses where there will be more mathematical discussions will be there. But you have not to lose something here. If you are more mathematically inclined, even then you can actually enjoy this course because there will be some programming tasks given in the coming weeks. So where you will build simple model, you will analyze those models, right? So all these things you will do. So if you have more mathematical inclination also, you will be able to enjoy it. So before, as I do not see any questions till now, so let me go forward and discuss certain key points that are most important if you want to learn this subject, as well as if you are preparing for the assignment and the final exam, what are the points where you should focus more about, right? So let me share my screen so that you can see and I can write there. I hope people can see my screen now. Okay. So what I have jotted down here is that key points of week 1 lectures. I hope you have at least seen some of those videos and listened to some of those lectures, right? So in these lectures, what we are trying to do is that there are some key issues that we have to keep in mind. For example, every mathematical model that you create, it does not matter whether it is a mathematical model, ODE based, or any model you are creating, the goal should be that it should be simple. Let me choose the color so that for my pen. So the first thing is create models that are as simple as possible. Because remember, I will give you one example. Suppose I am writing a model for something where I have written down a differential equation, dx dt equal to k1x plus k2 x square, something like that. Now remember, these are parameters, right? K1, K2, these are constants, right? Both are constant. So if you have to analyze this model, you have to know the value of this, right? So the bigger model you make, the more complex model you create, you will get trapped into the problem that you will not find the numerical values. You will not have an idea about this value of this constant. How will you analyze it? So we have to always make models which are simple, but still they can represent the dynamics that we want to capture. There should be meaning. They should not be meaningless completely. There's physical meaning, biological meaning should be there, but they should be as simple and smaller as possible. Now, the second point that you have to understand in this first week's course is that every ODE based model, whatever you are creating, you are actually using ODE, one object. If you are using one ODE, then it is one ODE based model. If you are using more than one ODE, it is an ODE system, right? System of ODEs, right? So any of these ODEs will represent a rate. Something is changing with time. That rate you are measuring, right? That you have to understand clearly. Now, before you jumpstart into any ODE-based model, you have to understand two key assumptions. And these two key assumptions are discussed in details in the lecture 2. Please follow that and try to understand that. One key assumption is that the system is homogeneous. Homogeneous means if this whole house, the room that I am sitting right now, if this is a cell and then molecules are jiggling around, then if I sample anywhere, I take a bucket and just scoop out some sample, right? Then I do the scooping somewhere else. So across this whole room, everywhere if I scoop, I should get similar type of molecules in similar numbers. That means all the molecules are homogeneously distributed, right? The problem starts if there is, unequal distribution. For example, suppose I am working on a cell in which I have a region. Suppose this is a region where certain particular membrane structure or something may be there for which some protein get more concentrated, whereas the concentration of the same protein somewhere else in the cell are lesser. If you are creating an ecological model, then suppose you have an organism which is a variable in your model and you are creating the model for that, and then this variable, this particular organism does not remain dispersed throughout the area of your model, throughout the system, but in a niche area they stay most. Then the problem is it is not homogeneous, right? Because depending upon the space, the variable, the dependent variable, depending upon the space, this distribution is varying. So in that case, what I have to do, I have to use PDE based model. So if I have a heterogeneity, spatial heterogeneity, then I have to use PDE. But in this course, we are not dealing with PDEs, right? So in this course, it is purely on ordinary differential equation and lots of model in biology with simple basic assumptions. We can actually convert the homogeneous system. So the system has to be well mixed. Another way of saying is well mixed. The second point that we have to remember, the system is very large, right? And I have given a very good example in the lecture 1 to explain why the system size has to be large. That means number of molecules involved in the system, number of people in your epidemic model, number of animals in your ecological model, number of cells in your model for an organ system. These numbers should be very large. If you have small numbers, less number, then what will happen when the number is less? When number is less, you get stochasticity or randomness. Yes, if you have followed biology literature, you may be aware that there are certain processes which are stochastic. If you look at single cell level, expression of a gene is a stochastic or a random process, and we know that. And we have beautiful mathematical way to probabilistically capture that stochastic or random behavior also. But even then, I can actually model a gene expression, translation, transcription, all these things using ODE, because remember, in most of the time, you are not dealing with a single cell. Very few people will do single cell experiment. Very few data is actually single cell data. Most of the textbooks that we have read to learn biology are coming from what we say is, all cell clubbed together and broken down, molecules isolated from that, and from there you get the idea how those molecules work. So when you do real-time PCR, RT-PCR, you are not taking one single cell, you are taking a 1 million cells, suppose, right? When you are doing Western blot, SDS page, you are not taking one single cell and isolating protein, you are taking millions of cells and doing the assay. So your data is coming from a large system. So all the individual cell will have stochasticity or randomness. As a whole, the system can be handled using ODE and can be considered as deterministic. Then another important thing that you should read and be very careful about, law of mass action. Here you should know how to write a ODE using law of mass action. In the lecture I have explained it. And this is a part where I have seen over time that learners may make mistakes, right? So be careful, try to understand how. I am sure in your undergraduate courses somewhere you have studied law of mass action. Please recapitulate that and follow the lecture and learn how to write differential equations, ODEs, using law of mass action. Although I have said in the lecture and I will reiterate, most of the biological processes that you will model are not following pure law of mass action, because most of the processes are not elementary processes. Those are a combination of multiple processes, and the concept of rate limiting steps are there. You may have studied in biochemistry, but still we take the law of mass action as metaphor and create models. So you have to understand how to write ODEs based on law of mass action, right? The third lecture you may have noticed is based on disease model. A very simple model, right? That has been given to you so that you can warm up. You can learn how to start creating model, how to integrate an ODE. But here in this lecture, you should focus on two things. First, try to understand, and I have tried to explain in this lecture, try to understand how we are using these two assumptions, that a system has to be homogeneous and the system has to be large. These two assumptions are utilized properly when we are building the disease model. So try to understand that, because every time you create a new model, you have to use these two assumptions and you have to use this appropriately, right? So try to learn that from this simple model. And the second thing you should learn in this, and you should practice this, is actually how to integrate and solve ODE. Now here some of you may think that, okay, do I have to learn the whole integration, complicated calculus and all these things? No. No, because most of the time in real life problem that you will deal in biology to model, those you will not be able to solve using the standard integration method that we have learned in the undergraduate course or even in 12th standard, right? Because those are mostly non-linear coupled ODEs and most of them are not solvable analytically. But still, I will insist that learn simple integration, very simple integration, right? Which we are going through in this course. And I have not given integration for complicated things. It's very simple integration, very simple differentiation. And if you don't have a book of calculus, it does not matter. You just search in Google, you will find tables where those simple rules of differentiation and integrations are given. Just learn those. Those are good enough. And if you are finding it difficult, there are a beautiful online tool. Just search for online differentiation, on different calculator for differentiation, calculator for integration. You will find online tool where you can do simple integration and differentiation. To start with, you can use this. But I will insist that why don't you try? Why don't you try, if you know basics of calculus, why don't you try simple integration, simple differentiation? You don't need to go into complicated, right? The second thing is that let us go to the other model, the fourth lecture, where we use the population model. What you should focus on is, again, in this problem, you should learn how we are doing the integration. It's a very simple integration, not a complicated one. Again, that will help you to learn how to do simple integration to solve a ODE. But you should learn one important thing apart from that in this population model lecture, and that is logistic function. Logistic function is a exponential, sorry, a sigmoidal function. By sigmoidal function, we mean it looks like a S-shape, sigmoid, right? And there can be many mathematical functions which can give sigmoidal function. So in this case, we are using a logistic function. What is a logistic function? Logistic function, if you carefully listen to that lecture, if you have the slides, you can open and check. A logistic function is something like this. It will be 1 minus x by k into x. So x is the variable, k is a constant. In the population model, k is called carrying capacity. But you can use this logistic function in many places, not just population. You have some behavior, some dynamics where you have got the sigmoidal behavior. And one very useful function in that case is logistic function. And many a time we plug that function to create a model. So try to understand it and try to understand its behavior. Simply, there are, if you don't have if you don't want to write a code or something, there are beautiful online plotters, right? Search for online plotter, or even in Google search window, simply type this one, 1 minus x by k, put a value of k, right? Suppose k is 10 or k equal to 100 into x and search. It will plot. So plot this curve and see how its behavior changes as you change k, right? That will give you an idea how this sigmoid function behaves. Okay, till now there is no question as I see. So I will go to the other important thing that you have to learn very carefully for assignment and exams for this particular course is that you should, I will insist, let me clean it a bit so that it becomes better. Okay, so this one, Euler's method, please practice it. You don't need to take very complicated functions. Take very simple thing. Dx dt equal to x. Take dx dt equal to 10 minus x. Create your own problem. There is a problem given in the what do we say, the on the lecture, but you create your own and then use Euler's method. We have already uploaded one Excel sheet where you can use Excel formulas to solve using Euler method. Practice that also. But what I want is that you should be able to use this Euler method by paper and pen yourself, and it is not difficult. It is school level algebra and any arithmetic essentially, and anybody can do it. Once you learn it, then you will realize the strength of it. Remember, Euler's method is 300 years old. But even now, people use Euler's method, particularly if you are doing very large-scale simulation for ODE-based model. Although it has error, when you do it by paper and pen, you yourself will realize where the error can come, the problems that we have been discussed in lectures. You can realize that where the problem is coming. But at the same time, you will realize the strength of it also. So please practice it by paper and pen. Also for exams and quizzes and everywhere, you should also learn this one by heart, right? Runge-Kutta method, you may not need to know details of the mathematics of it. Just try to grasp the idea that why you will need Runge-Kutta method. Because Euler's method uses one slope and it gives wrong answer for, approximation is bad if you have very sharp rise in the function. Whereas Runge-Kutta method can correct that to some extent, right? Because it uses multiple slopes. So you have to understand what is the advantages of Runge-Kutta method over Euler's method. That you should understand, right? And you should also understand the limitations of that. So you have to understand advantages and you have to understand disadvantages. All have been discussed in the lecture. So you have not to go anywhere. You don't need any textbook to go into it. Just follow. When you are listening, watching the video, just focus on these two. And when you are doing that, remember to understand why step size is so important. And that's why I am saying use paper and pen. Use the same equation. Suppose you want to solve this dx dt equal to 10 minus x yourself. for an initial condition given, you are using paper and pen doing it. Try it. Initially t delta t equal to 1, time state is 1. Then try t equal to 0.1 and see how the result is varying. That will give you idea, that will give you confidence that why this time state is so important, right? And that is important when you are creating models, right? If you are creating models, you have to understand the effect of the step size, and otherwise your model can give rise to a wrong result. Also, the last thing in lecture 6 that you have to understand is the utility of adaptive step size. Adaptive step size, fine? So in adaptive step size, what you are doing, you are not using delta t constant, right? For example, just now when I say use delta t equal to 1, so for whole solution you are using delta t equal to. Then I said, okay, now change delta t to 0.1. So from beginning to end you are using 0.1. So delta t is constant. Whereas in adaptive time step algorithm, you are changing it depending upon where you are in the function, right? So it is explained in the lecture. Please try to grasp the meaning, the logic of it. Because most of the modern algorithms which are implemented in softwares as well as libraries are using adaptive step size, right? Most of the time, right? So you have to understand the basic principle of that so that you can use them adequately and properly in the library, right? So these are the all few key points that one should keep in mind when you are going through this week's lecture. I think there is a question. Let me take that before I proceed. Can I use dynamical model for studying regenerative processes exclusively? For example, limb regeneration in salamander. Okay, Monisha, it's an interesting question. What you are asking, if I understand properly, you want to create a dynamical model to create how limb regeneration. So essentially, tissue formation, if I understand it properly, right? So there are dynamical models. In fact, there are dynamical models, and let me give one example, slightly deviated from what Manisha has asked. I believe most of you have heard of the name of Alan Turing, right? Alan Turing, famous for cracking the German code during World War. He was a British. He cracked the hidden code for German secret services, right? And that's how he actually saved us from the war, right? And also he was a famous for mathematician, because all the computers that I'm using right now, those are called Turing machine, because he defined how a computer should work, right? He developed mathematical idea. But many times most people doesn't know that Alan Turing also has a pioneering work which is called chemical morphogenesis. What is, if you're a biology student, you must be knowing what is morphogenesis, right? Genesis, maybe I'm writing wrong spelling, whatever it is. Morphogenesis, the formation of different cell type and from their structure in the body. We are all metazoan, we have multiple cell type, but we all originated from one single cell, right? That is miraculous, and it is happening every moment, rightly, right? That's why we exist, you, me, everybody exists. So now morphogenesis is another thing, another very interesting problem you may have seen. Apart from this, the embryonic development is definitely a very interesting morphogenesis. Apart from that, think about it, the formation of colors on the body of animals, right? Patches and all these things, dogs, your tiger and all these things, zebra. So Alan Turing gave a model called Turing's model, which is based on very simple thing. I have two molecules, A and B, and they interact with each other. One activate, another one inhibit, something like that, if I am not wrong. And they are diffusing, they are not in one place. In an area, they are diffusing. They are all jiggling around. And now he created a PDE based model. Remember, these are diffusing. So there is the issue of space, right? So when something diffuse, there is spatial heterogeneity. It is not homogeneous. Homogeneous means diffusion has stopped. Everything is homogeneous. So he created a partial differential equation based model, which if you simulate can give rise to beautiful patterns. And that has been widely utilized to understand morphogenesis, although Turing's original model may not be useful to create a model like as Monisha asked for the regeneration of salamander or something, right? But people do that type of dynamical model to understand how tissues are formed, how multiple cell type originate from one single cell type, multiple different cell type will originate, and they will organize with each other. So in this course, we will not deal with PDE based thing, but we will deal with one thing that you may enjoy, which is the first step in embryonic development. Obviously, one apart from this organization of cells to create tissues and then organ, then you and me, the multiple cell type has to originate from one single cell, right? You call them pluripotent stem cell, right? From there, they divide and create different other stem cell, and ultimately the adult cell, right? Those terminal cells you get, right? Which does not divide, does not differentiate anymore. So how come one cell type, suppose I have a cell type A, can give rise to cell type B and cell type C? How come? So there are beautiful mathematical theory that is called bifurcation, and we will learn this in our course in coming weeks. Not immediately now, but we will learn it. What is bifurcation and how it can be used during embryogenesis to understand origin of different cell type from one single cell. Monisha, I hope I could answer your question largely. Yes, it can be created, but most of the time people use OD based, PD based model. Is there any more questions? Okay, so as I do not see any more question right now, let me move to some example problem that we'll discuss here and solve, right? That will help you in your quiz, assignment and final exam maybe. So in the first lecture, I have discussed about creating, simple graphical models, right? So why do you need graphical model? Graphical model, you must have seen hundreds of graphical models actually in any cell biology, biochemistry. We use them because they can distill a large dispersed information in a simple way, and you can understand it easily, right? So when you are building a mathematical model, it is always good if you can draw the graphical model. If it is a very large system, there is no meaning of drawing the whole thing, right? But if it is a small system, 10 molecule, 5 molecule, you try to create a graphical. So here in this problem, if I zoom it bigger, a graphical model has been shown, right? I have two reversible arrow between A and B, and then X and Y has arrows putting on those reversible things. The question is in this system, what is the function of X? X is working as an enzyme, substrate, a product, or a component. Can someone who is present answer this? I can wait for 1 minute. The question is, if you can see this is graphical model is shown here. The question is, what is the purpose of X here? Is the X working as an enzyme, or a substrate, or a product, or a component? Can someone answer? Yes, Monisha, you are right. It is an engine. And now try to understand why we are writing this in this way. See, we could have written drawn this one in this way, like A plus X B, right? We could have done that. But remember, X is an engine. That's what we want to mean, right? Now remember, unless you are creating an explicit model. By explicit model, I mean the enzymatic reaction is happening where every intermediate step you want to show. If you want to do that, then your model becomes bigger. And remember, I started with saying keep model simple and useful. So most of the time, we do not show this intermediate step of reaction, right? What we are showing, A is converted to B with the help of the enzyme X. Now, if I write in this way, the way I have written by hand here, The problem is when I will write dx dt, the change in concentration of X, the enzyme, I may somehow consider this reaction where X is a substrate. So if X is a substrate or reactant, then X will get consumed, right? So this reaction should come into this dx dt equation. But the fact is I know X is enzyme. So in this reaction, in this particular reaction, X does not get used up, its amount is conserved. So that means these reactions should not be represented in my dx dt. To avoid this confusion, we put arrows, right? The arrows are drawn in such a way, as you can see here, the arrow is from X and it goes and hit the reaction path, right? And it tells me, arrowhead is there, it tells me X is positively regulated. X is positively regulating. So it's an enzyme which is positively regulating this reaction and it does not get affected by that. Its concentration does not change, right? If X is inhibiting, then what I will do? I will show a hammer. Again, X is not actually getting used up by this inhibition process. Only it is inhibiting. So it is a control. It is a transfer of information. So that is how we show, and the answer here will be enzyme. Yes, Bharat, I will say what is compartment, right? Compartment is very simple. Suppose this is a cell and you have considered explicit compartment, right? All cell has some compartment, you have to understand. Although we say that in bacteria we do not have any membrane bound compartment, but even if you do experiment, you can see actually, people have observed under microscope also, there are zones where some molecules are more, there are zones in the cytoplasm where some molecules are absent, something like that. So suppose you take a eukaryotic cell, a human cell or something, so you will have compartment, like you maybe have a Golgi here, right? You have a nucleus here. So these are compartments, right? Separate compartment, right? So a molecules can move from one compartment to another compartment. And I have a next question to based on that. So I hope you will be able to understand what I mean by compartment specifically. So let us go to the next question. See what we have shown here. The problem says identify the mistake in the graphical model given. Just to tell to Bharat to specify, this system has two compartments. Two compartments. Which one? This one, the bigger outer ellipse is one compartment, suppose one, and this inner one, suppose that is nucleus, that is second one. So I have two compartments, right? I can assume both the compartment are homogeneous, meaning compartment one everything is well mixed. I can consider compartment which is completely well mixed. Let me give you another example of compartment. Suppose if you are a student from pharmacy background, you should know. Suppose you are making a model for pharmacodynamics. How the drug, I have taken a swallowed a drug. It has gone to my gut. It is dissolved there. Now through blood it is getting distributed through my body. So you can consider each of the organs. Suppose this is brain. Then suppose I have a liver. Then I have suppose skeletal part as another organ system you consider, right? And you have the gut. So from gut, maybe the drug has gone from liver to the hepatic circulation. From that it has come here. From the suppose it will go to heart, obviously heart maybe, and it got distributed like this. So now you want to study the dynamics of this, right? So each of these organs are now compartment in your body, right? So hope the idea of compartment is clear. So now look back to this problem. So in this problem, what do you think is a major mistake? If the idea of compartment is clear, can someone answer? What is the major mistake in this problem diagram? Bharat, if you are there, can you try now to look into this diagram and say what is the problem in this graphical model diagram that I have drawn? What is the mistake? Okay, if it is not coming, so let me give you the answer. It's not so tricky. See, I have A. A is becoming B, and B is big, which I marked as BC, because suppose C is representing cytoplasm, so B subscript C, and that B diffuses into nucleus, suppose the second compartment, and that's where I have marked B by 2 variables. It's the same molecule B, but when it is in B cytoplasm, I have marked it as BC, so that I can keep a track how many of B are in cytoplasm and which is in my nucleus. I have marked this as new variable Bn. Summation of these two will be equal to total that in the system. So suppose 200 molecules of B are present in the cell, 10 are present in the nucleus, 190 right now are present in cytoplasm. Just now 2 molecules diffuse into nucleus, so now there is 12 in the nucleus, 188 in the cytoplasm. So I can keep accounting both the way, right? So if I am doing that, that is the right way to do that. Then why I am showing BC again here? The Bn is coming out and become to the outer compartment, right? So this BC you don't need to show separately. I can simply put an arrow, right? I can put an arrow showing that the Bn to BC is coming. Something like this, right? So I can use a double arrow. I don't need to use twice, right? So I do not need, the answer is BC should be shown only once in the same compound, right? So we have to show BC. Yes, Manisha, the second answer is correct, that we have to show BC only once in the compound. Good try. So now let me go to our next question. The question reads that we have created a logistic growth model. Again, remember, logistic growth model is sigmoidal, right? It rose and reaches and saturates. We have the population model lecture, we have discussed logistic growth. And the fishes growing in a pond. For a fish growing in a pond, we have assumed that no fish dies. So they are immortal, right? That makes our life in modeling easy. And the carrying capacity of the pond is 2000. What will be the population size of fishes in that pond after a very long time? So if I remember the equation, or if you don't remember, you can look into the slides for this lecture. Suppose x is the population size, then dx dt, I will have some constant k, suppose, right, rate constant k, and then it's the logistic growth, so I have x divided by c, c is the carrying capacity into x, something like that should be there, if I remember correct. Otherwise, this is logistic function, I can write it. There should be no negative term or any other term afterward because there is no death. Now C is given as 2000. So remember what we are asking. We are asking what will be the population size of fishes in the pond after a very long time. This is the tricky thing in mathematical analysis. What do you mean by very long? Essentially we mean it tends to infinity. So that means I am asking you t tends to infinity. Right? Now, how will you approach this problem? You can approach it in two ways. One is bit laborious, another one is bit smart. So the laborious way, let me tell you. The laborious way is to actually nothing but doing dx 1 minus x by c x equal to kt. You write rearrange term and then you integrate both sides. And you get x as a function of time, and that has been done in the lecture. If you are lazy, you can look into the lecture and you will find how to do the integration and you will get the answer. If you want to practice the integration, practice it here for this problem. But remember, that is a laborious way of doing it. I will not approach this question if in my exam I have got 2 minutes only to do it. I will not do the integration to solve this. I will use some common sense. What is that common sense? Common sense says that logistic growth has a sigmoidal function. right? So that means my sigmoid will look like something like this. It should start somewhere and then it will saturate. Why it will saturate? Let me look into the equation. My equation is K into 1 minus x by C into x. So when x is equal to C, when x is equal to C, this whole thing is equal to 1. Remember, this is equal to dx dt, right? So when this is 1, x by C equal to 1, that means x equal to C, then dx dt is 0. That means in this saturation, x is equal to C. So this is the, if it is the x-axis, this is time axis, then this is C. So saturation will be at C carrying capacity. That's what a logistic growth function says, as simple as that. Whatever purpose you use, it does not matter. And when the saturation will come, I do not know when the saturation will come. I do not know what is this time. I do not know this time. But I am sure that if I wait eternally for eternity, means I am waiting for infinite time, very long time, surely the system will reach saturation, right? And that gives me the answer. So the answer will be simply the carrying capacity. What is the carrying capacity? Carrying capacity is 2000. So as the function is sigmoidal, if I wait for a long period of time, t tends to infinity, then obviously by infinite time, the system should saturate. And where it should saturate? This is a sigmoid function of logistic growth function. Logistic growth function saturate as at the carrying capacity. The carrying capacity is 2000, so my answer should be 2000. Right? So this is a shortcut. So when you are answering question, just try to be a bit more smarter, right? You may avoid actually lots of complicated thing. And as a student, I always follow one simple thing. I look into the marks given to the question. If the mark is big, then it may require lots of calculation. If the marks is very less, 1, 2, that means there must be some smarter way to answer that question. I don't need to go into detailed calculation, right? The same thing. Okay, let me look into another problem before we wrap up, maybe after some time. Is there any question about till now about all the problems that I discussed? Okay, so let us look into this problem. We will learn to model degradation of protein, degradation of mRNA, death of cell, and all these things. But even then, before we learn that in the lecture, here is the problem where we are saying degradation of a protein is proportional to the current concentration of the protein. That means the rate of degradation, obviously, although it is not written here, right? So the rate of degradation is proportional to the concentration, right? Now the question is, which of the following ODs, some 4 ODs are listed here, is suitable for modeling this process, where the concentration is actually in the square bracket and K is a rate constant. So I think it's very easy to answer. If you simply in your college level, if you have done first order reaction, second order reaction, or even if you have understood or remember the law of mass action, you should be able to tell. Can someone try? Which one will be the correct answer? 1, 2, 3, or 4? Okay. Yes, Manish. Manish, Anand, Manisha, both of you are correct. The second one is the correct answer. Second one is the correct answer. So just to explain to other, so I will write dp dt. This is my rate, and this is proportional to P. This is called first order reaction. Remember, first order. I am borrowing for chemistry, right? Chemical kinetics. First order reaction. If it has been second order, then I would be P square or P into Q, something like that. And to remove this proportional symbol, if I want to replace with equal to sign, I have to put a constant term. So that constant term will be K. But be careful. I have to put a minus sign, right? Because it is decay, right? It's decay. So the concentration of P is decreasing. I should not leave positive sign there. I have to put explicitly minus sign there, right? Because then only it will represent the decay or degradation or death, right? So I have to be very careful. I have to explicitly put minus sign. Good. Both of you have done right answer. Thanks for commenting. Let me look into another problem. I think we have a couple of minutes time also. Okay. Look into this problem. Growth of yeast in a fermenter is modeled using the following code. dP dt is equal to R into P minus D into P. Right? Here P is the population size, R is the rate constant for growth, D is the rate constant from death. Initial population was P0. What will be the population size at time t? Right? So, it is a practice of integration with bit of smart trick. What should I do? dP dt, I write, right? So do someone want to try which one will be answer? I have 4 answers given here. 1, 2, 3, 4. Hope you people can see. I'll wait 1 minute if you want to answer. Very simple. Manish, you said 1. Right. Bharat, you said 1. Right. Correct. Both of you are correct. See, again, remember, this is a question where you don't actually need a 1 page long integration, right? This must be something simple. It's my MCQ, right? So the trick is very simple. It is actually trying to confuse you, because what you have here, R minus D into P. And just now we have done other problem where we have said population change is equal to K into X, right? The basic what we call Malthusian growth, exponential growth will happen. And in one lecture we have discussed that, right? And that one comes that X at time t is equal to X at time 0 e to the power K into T. This integration you have done earlier. So here this is K. That means T should be T0 e to the power what is K? K is R minus D into T. As simple as that. Again, remember, this course is for a broad audience. Some of you are good in calculus, some may not be so comfortable with calculus. So we are keeping this integration, this differentiation bit simpler one. But if you are really interested in this topic and you are comfortable with calculus, please try to create modules which you, and there are lots of modules, you can get literature anywhere, right? Try to solve those equations. Try if this is one single OD, not for a couple system. Don't try it, you will waste your time. If it's a single OD, try to integrate it. If you get stuck, okay, use an online tool to solve it, right? That will, warm you up for more to learn, and a very interesting thing can come out of it. Otherwise, for this course, you don't need to go into complicated integration and differentiation. You just need to know the basics of this, right? Okay, so these are all the questions that I have brought today so that we can discuss with you. If anybody has any query, I am ready to accept. Please come on. You can write your question here. Any query regarding the course? Just to remind you, everybody, he present here. One student has posted earlier one question that the reading material, the textbook. Remember, all the textbook that we mention in the course are, or paper sometime we mention, and in the course material or below each lecture you may find some paper or books are there mentioned, right? So those are not compulsory. Those will not change your score in assignment or exam. Those I have given because if you are hungry to know more for that, right? So for example, suppose we are learning, you may see in one video that we are learning how ligand receptor binds. So there is a very classic paper on that. So I have given a link to that. It's a free paper, you can download and read that. But that's not part of the course. That is given to you so that if you are you have the hunger to learn more and more, and you're excited to learn more, you can go for that, right? Otherwise, if you simply follow this course, do the assignment, you should be sailing through smoothly to this course, even in the final examination. I can assure you. If you are struggling with something, post the question in forum. If you if you don't want to post there, if you want that you should write me a personal mail, then write to my official email ID biplabbose@iitg.ac.in, right? So I'll be able to help you, right? And remember to join these live sessions where you can clarify your queries, right? If you do this, remember this course will be very entertaining and you will sail through the exam very nicely, smooth. Any question? So if you have any more question, please post it, because otherwise within 2-3 minutes we will end the session. Okay, Bharat, you are asking, in a population, how can we model starvation, right? Okay. If I want to keep the model simple, and what happens in case of starvation, that can create a trouble in your problem. The simplest one, I can assume that if you have starvation, the cells will die. Suppose you are modeling cell. Take it easy. Let us not jump into a starvation of human being right now. Let us think about starvation of cells, or suppose, okay, some animals may be starvation. Keep it, suppose make it more easy, bacteria or some microorganism, and there is some amount of limitation of resources, right? Then what will happen because of starvation? There can be many things that can happen in starvation. For example, if you have heard of dictyostelium is a unicellular organism, but when they starve, they come and become one clump of cells, right? And they become a multicellular organism. So I don't want to go into those type of model now. Suppose simple thing, if there is starvation, there will be death. Or if there is starvation, there will be cell division will reduce, right? Now, what you can do is actually you can incorporate that effect of starvation in the rate constant for death. You can do that, right? Now you can make, what you can make is that you can actually make some function. Suppose, for example, if you are writing dx dt, just only the death part I am writing, right? Minus kx will be where the death rate is constant. Now you want to make this a function of population itself, because your starvation may depend upon population computation, right? So here also you can use some sort of sigmoid function, which will make, as x will increase, this k will become bigger and bigger and bigger. That means as more and more population increases, your rate of death will also increase, because people are, the organisms are starving because there is shortage of food, right? So this type of modification you can do. So you can incorporate the effect of starvation, which can be cell density dependent, population dependent, either in the death or in the birth rate constant. So you can make the birth rate constant or the death rate constant dependent upon the population base, right? Hope that answers your question. Okay, is there any more question? Anyone? Okay, I think we have almost at the end of almost 1 hour done. We will, let me thank all the participants who participated today in this live session. Thank you for with us, and particularly for those who asked questions, interacted, gave the answers. Please join back again next week in our live session. Email will go, notification will go, announcement will go. We'll have another live session in the next week and also in the coming week. So please join this live session and post your questions and try to answer the practice problem that I'm discussing in this live session, so that you can understand the nitty-gritty. And if you have any difficulty, you can share that with me so that I can explain them and other people also, because many times maybe which is difficult to you is also difficult to other people also. So please join these live sessions, and thank you once again for joining today's live session. Thank you all. We'll wrap up now.